Hello, good afternoon, and welcome to the this year's Jean Bannister Prize Lecture. So our Jean Bannister Award is an annual award that was established in 2016 in memory of Professor Rachel Jean Bannister, who passed away in 2013. The prize supports a series of lectures given by early career scientists in any field of physiology. So this year's lecturer is Dr. Sylvia Schröder. She is a Sir Henry Dale Fellow and Group Leader at the University of Sussex. And her research focuses on how behavior shapes visual processing in the early visuals system. She did a postdoc at UCL in the labs of Matteo Carandini and Kenneth Harris, and she made key discoveries showing that neural signals from the retina are influenced by behavioral states such as locomotion and alertness. A surprise discovery. She uses powerful tools such as two-photon imaging and NeuroPixels probes and optogenetics in mouse models to investigate complex brain behavior interactions. She got her PhD at the ETH in Zurich in the lab of Kevin Martin, where she studied the functional architecture of the primary visual cortex. She has a master's in neural systems and neural computation from the University of Zurich and a bachelor's in cognitive science from the University of Osnabrück, where she started exploring attention, saliency, and task-driven perception. Okay, so with no further ado, I'll hand over to Sylvia to deliver the our Jean Bannister Prize lecture.
Thank you very much for this introduction and for inviting me. Yes, so as you can see here on this slide, let me just get the laser pointer on. Right. So in my lab, we use the mouse as a model system to study the neural processes underlying vision and specifically the first stages of visual processing. And here you can see, for example, axons of the retina on their way from the eye to the thalamus and then further on to other brain areas. So in case you're not studying vision, you may ask yourself, well, what is there still to find out in this field? Since almost 100 years now, we study visual processing at the level of single neurons, and any AI tool, even on your mobile, can now tell a cat apart from a dog from a photo and can even track single objects in really crowded scenes. But I think there are a few things that we still haven't understood. And one of those is the question of how behavior and other non-visual contexts in general affect visual processing. So let me start with a quick overview of a simplified textbook version of what we know about the early visual system. So here you see the two main cell types in the retina. We have the on-center retinal cell, which responds really strongly if there is a bright spot in the center of its receptive field. And we have an off-center retinal ganglion cell type, which responds most strongly with a dark spot in the center of its receptive field. And then later, neurons in the cortex integrate information from these cells that have circular receptive fields to create something like an edge detector, which responds specifically to a certain orientation, as you can see here. So this was discovered in the 60s in the last century. But even just 10 or 20 years later, people realized that it's not just the visual input that influences activity in the early visual system, but also the state. So, for example, sleep or arousal. So here you see data that was recorded in a drowsy cat. You can see here the EEG recording, which shows that in the beginning of this recording, the cat was more awake, and here it became more sleepy. Here are the recordings of multi-unit activity in response to a spot of a stimulus. And if you align the activity to the onset of these stimuli, you can see that the activity is larger in the thalamus when the cat is awake rather than when it is asleep. And similar results were found in primary visual cortex. Again, you have the EEG here. In that case, the cat was more sleepy in the beginning and then became more awake later on. The stimulus here was a drifting bar stimulus, and you can see again that the single cell responses were larger when the animal was awake compared to sleepy. Okay. But if you want to study behavior and specifically locomotion, the cat as a model is really difficult to use. So the question of how behavior and locomotion impacts visual processing really only caught traction once the field switched to the mouse as a model and developed ways to record neural activity in awake rodents that could run relatively freely. And one important invention was this device here, where you can head fix an animal, which is then voluntarily running on top of a styrofoam ball, which can move in any direction, and the movement of this ball is recorded. So soon after this was invented successfully, people then started looking at how visual activity here in primary visual cortex of the mouse is affected by locomotion. They used the common stimulus of a drifting grating. Here you see one drifting in this direction. The drift direction changes in every trial. And you can see here in this example of the response of a single neuron, it nicely responds to the oscillations of the grating stimulus, but the response is larger when the animal is moving. And here you see all the response amplitude summarized in the direction tuning curve. In red, you can see the responses to the different directions when the animal is stationary, and in blue when the animal is moving. So with this historical background, now I want to tell you what we are interested in the lab, and we focus specifically on 3 different non-visual contexts, which each pose a different challenge or demand on the visual system. We have locomotion here, which completely changes the visual input statistics. We look at the effects of pupil-linked arousal, which may change the importance of some visual inputs, and also the effects of reward, which marks relevant stimuli and features. And we're particularly interested in where the information of context and vision is integrated in the brain, how the activity, the visual activity is modulated, and what the purpose of this integration is. And what I hope to convince you today is that this integration between context and visual signals happened much earlier than was previously thought, and that this integration makes coding more efficient and maybe behaviorally relevant to the animal. The main part of my talk will focus on the impact of locomotion, so let me tell you a bit more about why this is an interesting question. So when we walk, our visual input changes completely. Suddenly, almost the whole visual scene flows by, while it was almost completely stationary when we're not moving. In that scenario, our brain needs to solve 2 problems. It needs to process completely different statistics of the visual input, namely faster moving visual input, and it also needs to distinguish between self-generated visual motion from motion that is caused by movement of others. So to do this, the brain could solve these tasks either by simply processing the incoming visual signals, or it could use information about its own behavior. So for example, I am walking and therefore I expect faster visual input, and therefore I tune my visual system to these expectations. And the questions we are asking is there a direct link and impact of behavior on visual processing, preparing the brain for the change in visual inputs? To ask this question, we focus on one particular brain area called the superior colliculus, which exists in all vertebrates, including the mouse and humans. And the superior colliculus, together with the dorsolateral geniculate nucleus in the thalamus, these are the 2 only brain areas that receive direct retinal input that has image-forming information. In addition, the superior colliculus gets input from the primary visual cortex and other cortical areas. Here I'm focusing on the superficial layers of the superior colliculus, which are mainly visual. But the interesting thing about the superior colliculus is also that it then transforms the visual information that is received in its superficial layers into motor functions, for example, turning towards attractive stimuli like food or turning away from predators and threatening stimuli. So this gives us the possibility to test our hypothesis we come up with regarding visual processing on behavior quite directly. But how do we measure neural activity in the superior colliculus of awake mice? For this, we use two-photon imaging of a calcium indicator called GCAMP, which we virally express in the superior colliculus. And here you can see one coronal slice of the mouse brain from a common brain atlas of the mouse. And when I started this project and looked at this slice, I thought that's great, should be really easy to optically access these neurons, which are at the surface of the brain. What I hadn't appreciated and only realized when I started doing my first surgeries is that there is a really big blood vessel, a sinus in the dura, right on top of the superior colliculus. So it took me about half a year or so to figure out a proper surgery to get access to this area and then build an implant with which I could push away this big blood vessel towards the front of the brain. So I get optical access like here, that you can see here in this photo. And if everything goes well, then doing two-photon imaging, you can see images like this, where you can see single cell bodies that are expressing GCAMP in the brain. While we do these experiments, we are also tracking the pupil size and other behaviors of the animal, as well as its running speed. And like other people have seen, we also observed that the pupil size and the running speed is highly correlated with each other. And for simplicity, for the following analyses, I will just distinguish between two broad states, behavioral states, namely the quiet state, where the pupil is quite small and the animal is stationary, and the active state, where the animal is running and the pupil size is very big. And here you can see one of such recordings from neurons in the superior colliculus. Here you can see several neurons recorded simultaneously, and they were sorted by their correlation with running speed, which you can see here, as well as the pupil size. And you can clearly see, even though there was no visual information, so our monitors just showed a gray screen, you can clearly see that the top portion of these neurons here are positively correlated with running, while the bottom portion is negatively correlated. When we then started to show our grating stimuli, as you can see here, these tick marks show the onsets of the stimuli. We saw there is visual activity indeed, but this does not override the modulation by locomotion and internal state. Okay. Our next question was then how locomotion affects the visual responses in more detail. So as I said, we showed the gratings drifting in different directions, and for each trial we labeled it as either active when the mouse was running or quiet and the pupil was small. What you can see here is the responses of two example neurons. You can see the calcium traces in response to individual stimuli. In black, the response during the quiet state, and in red during the active state. And all responses are summarized here in the direction tuning curve. And you can clearly see that especially the response to the preferred direction of motion is much higher during the active state in this particular neuron. In general, the change of the responses can be well described with the linear relationship, as you can see here. Similarly, in this neuron, the responses are affected by locomotion, only that now the responses are actually reduced by locomotion, but again, the relationship is linear. So we saw similar things across the whole population, and what this linear relationship also means is that the preferred direction does not change between the states. So no matter whether it's quiet or active, the preferred direction is more or less the same. However, the direction selectivity actually is somewhat reduced during the active state. We also quantified how much the responses are changing by locomotion, and for this we took a very simple measure. simply the difference between the response during active minus quiet, and this was normalized by the mean response of these two states. And when we look at this for the whole population, we see in black that about 50% of all the neurons are significantly modulated by locomotion. And again, half of this population is upmodulated and the other half is downmodulated. Okay, so when we saw this, we were quite surprised because at that time we only knew that locomotion is affecting responses in primary visual cortex. There was hardly any information about subcortical areas and none for the superior colliculus. So the first question we asked then is, where could this information come from? And we thought, well, maybe it's simply inherited from primary visual cortex, which has a connection to the superior colliculus. So to answer that question, we did basically the same experiments, same gratings, same behavioral paradigm, but now we used electrophysiology to record neuroresponses throughout the superior colliculus. And in the control condition, everything was as normal, but in half of the other trials, we inactivated primary visual cortex using optogenetics so that the excitatory neurons in V1 that project to the superior colliculus are silenced. And here you see responses from one example neuron. In the control condition, as before, locomotion has an effect on the responses. In this case, responses are increased during locomotion. And then when we inactivated V1, the responses generally were reduced, but the influence of locomotion was still apparent. And we saw the same effect across the whole population of superior colliculus neurons we recorded. Here you see response modulation in the control condition and a similar picture when V1 was inactivated. So in conclusion, we can say that the effect of locomotion was not inherited directly from V1. So when we saw this result, we were wondering again whether it's actually the locomotion that has an effect on our visual responses or whether there's something else that has an effect. And we were thinking that maybe it's actually light exposure. So or if you remember at the beginning, I told you that when the animal is running, also the pupil is dilating. This means that more light is hitting the retina, which could influence the visual responses of the neurons. So we thought the best control experiment to show that this is not the case would be if we recorded activity from the retina while the animal is running or not running. So if we find that locomotion has no effect on retinal responses, then we can be sure it can't be simply the light effect. Okay, to do this, we used a trick because, as you can imagine, it's quite difficult to record retinal responses in an awake animal, in an awake mouse. So instead, we used this trick of recording calcium activity in the retinal axons inside the superior colliculus instead of inside the cell bodies. So we injected a version of a GCAM called side GCAM that has a higher concentration at the axon terminals to do these experiments. Here you can see a confocal image ex vivo. So you can see here in green the GCAM of the axon terminals in the superficial layers of the superior colliculus. Here's a zoomed in version, and in blue you can see the cell bodies inside the superior colliculus. And here's a typical image when you do 2-photon imaging of these retinal axons. You can see single terminals here. Exactly. These experiments are quite tricky because, especially if the animal is moving, the brain also moves, and it doesn't just move in X and Y, but it also moves up and down, so that your imaging plane is actually moving inside the brain. This is particularly tricky for such very small items like the axon terminals. So to get around this and get a very good signal, which has no artifacts of movement, we did not simply image a single plane, but imaged a whole volume. So basically a stack of several planes with very small distances across. And then at each time point, we can select the best matching plane that matched our reference plane. And so across time, we then always have the information from the same depth in the brain. And if everything is okay, you get movies like this. This one is sped up 10 times. You can see the activity of single retinal axon terminals. You can see that with this arrow, when the grating is on the monitor, it also shows you the direction of movement of the grating. And you can see that some retinal axon terminals actually respond mostly to the offset of the grating, and others respond to the grating itself, and they are tuned to specific directions of the grating. Okay, now, when we recorded these data, we realized our control experiment is actually not working out. We still saw effects of locomotion in the retinal responses. So instead, we now really need to make sure that there is an effect of locomotion without any visual input. So therefore, we put the mouse in complete darkness and did the recordings. And here you can see the activity again of several retinal boutons imaged simultaneously. Again, they were sorted by their correlation with the running speed. And you can see, to our surprise, that the activity of many retinal axon boutons are positively correlated or negatively correlated. Again, when we showed the grating stimuli, we saw clear visual responses, but still they did not override the modulation by locomotion. And as before, we asked, well, what's the effect of locomotion on the visual responses? And here you can see 3 example retinal boutons. And very similar to before, the response amplitudes were modulated by locomotion. If we look at the whole population, we see that the preferred direction is not changed. Direction selectivity goes down by the active state. And we can see that responses are up or down modulated, while in the retina, there's a slightly higher trend for a down modulation of the responses. And here's a direct comparison in this cumulative histogram between retinal boutons and the SC neurons. You can see in the retina, the effects are slightly weaker than in the neurons. And again, there's this trend towards a reduction of the responses. But probably there is this locomotion effect, which we see in the SC neurons, is already to some degree inherited from the retina. So now, as I said, this was a big surprise to us. And again, we asked, how is this possible? What's the possible mechanism? How can these retinal axons know about the state of the mouse? So we came up with 2 simple hypotheses. One is that there are neuromodulators that act directly on the axon terminals of the retinal axons and modify the calcium concentration inside, which we then would pick up through our imaging method. And the other possibility, which is possibly more exciting, is that somehow this behavioral information, maybe by neuromodulators, actually reaches the eye by so-called retinopetal axons that come from the brain to the eye. Okay. Now, with our imaging method, we were not able to distinguish between both hypotheses. So we were thinking of how we could test whether this hypothesis of the retinopetal axons is true. So one idea was to now use electrophysiology and record the activity directly in the axons on their way from the eye to the brain to see whether we already see a modulation of visual responses there. And this is what we did. Here you can see how we targeted with our NeoPixels probe the optic tract. And this is quite tricky because, as you can see, it's quite thin, so it's easy to miss. But in some cases, we hit it with our NeoPixel probes to convince ourselves that we're actually recording from retinal axons. We showed a relatively fast full flicker stimulus. And here in these two cases, you can see that the units could follow this fast flicker. We also mapped receptor fields, and as expected from retinal units, the receptor fields were relatively small. And we also checked the spike waveforms across our NeoPixels probes during stationary and running states to make sure that we're still recording from the same units during both states. And here are the results from two example axons. You can see here the running trace again, and in the second row you can see the firing rate of a single retinal axon. And also it's not very clear in this raw data. When you calculate the cross-correlogram between running speed and firing rate of retinal axons, you can see that this unit is significantly positively correlated with running speed. And here's a second example where we also found a positive correlation. Now, this does not mean that there are no neuromodulators acting on the retinal axons inside the superior colliculus, and my guess is that both mechanisms are at play here. Luckily, this finding was also confirmed by another group. So whereas we could only record from 25 units because of the tricky experiments, this group here recorded far more units and could confirm that indeed many of the retinal axons in the optic tract are significantly correlated with running. And another confirmation comes from a different group that recorded calcium activity again from retinal axons now in the thalamus. Here they used wide field calcium imaging, and like our data, they showed that arousal, which is highly correlated with running, affects the population activity of retinal axons in the thalamus. Okay. So now again, we had this question, how does the eye, without any visual information, know about the behavior of the animal? And this question was recently tackled by, again, another group, which showed that histaminergic neurons in the hypothalamus project to the retina. So here you can see these retinopetal axons, which really spread out across the whole retina. Also, there are very few axons of those. And this group could also show that histamine has an effect on the activity of single retinal ganglion cells. Here you see the activity before and after application of histamine. So there are possible mechanisms that could modulate the activity inside the retina. Okay, so now when I started my own lab at Sussex a few years ago, we wanted to know whether locomotion also has an effect on visual responses to other visual features other than just the direction of motion. So two members in my lab, Liad and Maria, they tested how locomotion affects tuning to temporal frequency. And here you can see one example of a neuron in the superior colliculus during the quiet and the active state. Here you see the tuning curve for temporal frequency. And as before, you can see that the response amplitude is changed with activity. But more interestingly, you can also see that the preferred temporal frequency changes towards a higher frequency. Okay. When we look at the whole population, and here we just looked at the response modulation, so how, the response amplitude changed. As before, for direction tuning, we saw that half of the modulated population increased the responses and half decreased it. But more interestingly, we saw here, or we compared the preferences for temporal frequency during the quiet and the active state. In blue, you see the neurons that were significantly changing their preferences, and most of the neurons actually changed towards a higher temporal frequency. And to kind of look at the combined effect of response modulation and preferred temporal frequency, we made another plot. Here you can see the preferred temporal frequency of the neurons during the quiet state and how their responses change. And you can see here that it's mostly the neurons that prefer the lower temporal frequencies that increase their responses during locomotion. And what I don't show here, but I can tell you, is that it's these neurons also that mostly increase their preferences for temporal frequencies. We tested the same thing for retinal axons inside the superior colliculus and basically found qualitatively the same effects, also to a weaker degree. So here you see that some of the retinal axons changed their temporal frequency preferences, but fewer did. But there is already an effect in the retina. So in conclusion, from this data, we can say that visual neurons in the retina and in the superior colliculus are better tuned to faster visual inputs during running, which makes a lot of sense. We also tested spatial frequency tuning. And again, in terms of response amplitudes, we saw some neurons are upmodulated, others are downmodulated. In terms of preferences, we also saw a very strong effect in the neurons of the superior colliculus, showing that running actually reduced the preference of the spatial frequency. So neurons now liked lower spatial frequencies. But there's no dependence between preference and change in the response amplitude. And again, qualitatively, but to a weaker degree, we see these changes in the retinal boutons. Now there is the question, why would the brain want to change the preferences for spatial frequency when the animal is running? Spatial frequencies do not really change or do not change at all when the animal is running. So why would you want to tune your neurons differently? So we think it has may actually have to do with tuning to visual speed again, because if you change a spatial frequency of the stimulus, or you reduce the spatial frequency of the stimulus while keeping the temporal frequency constant, what is happening actually is that the visual speed of the stimulus increases. So again, these neurons here, or these data show that the neurons are better tuned during running to higher visual speeds. Okay. And we're, again, not the only ones seeing these kinds of effects. So even in the Drosophila, people have seen that visual responses, or their tuning to temporal frequency specifically, changes. So you can see here a tuning curve, population tuning curve when the animal is walking. In general, they prefer higher temporal frequencies compared to when the animal is stationary. And even in humans, there is at least psychophysical evidence that something like this may be happening, because there are studies that show that optic flow appears slower when people are walking. And this is consistent with visual neurons responding or shifting their preferred temporal frequency to higher frequencies during walking. Again, possible effects of this could come from histamine. So the same group that showed this anatomical connection and also effects on response amplitude showed that histamine also shifts the preferences for temporal frequencies to higher frequencies in retinal ganglion cells. Another effect could come from the light exposure. So again, when the pupil enlarges, more light hits the retina, and this also shifts temporal frequency tuning curves towards higher frequencies. So far, we have preliminary evidence that this is not the only effect, because when we dilate the pupil and therefore keep the light exposure the same, we still see a shift in preferences for temporal frequencies. All right, so now I've shown you several effects of locomotion on visual responses, on visual tuning, but I also showed you that the effects across the population are quite varied. So we turn to another question now, asking whether state modulation or locomotion affects different functional cell types differently. And the idea was that if some cell types are modulated more than others, then this could point towards the purpose of this modulation. And to functionally classify the neurons in the superior colliculus as well as the retinal axons, we used a stimulus protocol, which is commonly used to classify retinal ganglion cells. Here it shows you the contrast or the light level of the stimulus on the monitor. So it switches here from black to white, back to black, and so on. And when we showed this stimulus to our neurons and axons and then used clever classification protocols, we found strong evidence for 8 different functional cell types. And here you can see the average activity of these 8 clusters. Okay. I should add that this is still preliminary data and research is in progress. But so far, we use these classifications to see how locomotion affects each of these cell classes. And the results you can see here. So let me walk you through this. Here on the x-axis, you can see the 8 different cell types, in purple for the neurons and in green for the retinal axons. Here you can see how the response amplitude changes, here how the temporal frequency changes, or how the spatial frequency changes here. So on first sight, there's really no clear pattern. It seems locomotion really affects all cell types relatively similarly. The only ones where we could see something maybe differently happening is this cell type, which looks like an on-off transient cell type, where responses are mostly down-modulated and temporal frequencies are shifted towards lower frequencies. And this type, which looks like an unsustained cell type, where the temporal frequencies are upmodulated, so shifted towards higher frequencies, and spatial preferences are shifted towards lower frequencies. But yeah, no super clear pattern, but we are still working on this. Okay. Now I want to tell you what 2 other members of the lab are looking at. This is Florencia and Rehan. And instead of just looking at tuning properties and how they're affected by locomotion, they look at how locomotion and the active state affects temporal dynamics of superior colliculus neurons. So for this, they used electrophysiology with neuropixel probes to get a much better or higher temporal resolution compared to the imaging. They recorded through all the layers of the superior colliculus and again used our drifting grating stimuli. Here you can see responses from 2 different neurons. This is a raster plot showing the spikes for several trials during the quiet state and here during the active state. And the firing rate is summarized across time here by these PSTHs. So you can see during the quiet state, the response is reduced, but during the active state, the early response is much higher, while the later response is much lower. And this reduction in the response across continuous visual stimulation is called adaptation, and it's thought to be efficient because it saves energy when the stimulus is not changing. And it also enables the neuron to respond strongly again when the stimulus changes. And we can see here in this example that adaptation is stronger during the active state, so it may better enable the neuron to respond stronger at a change of the stimulus. Here you see a different example with slightly different temporal dynamics, but again, the late response of this neuron is reduced during running. And here's a summary of what we saw across the whole population of neurons that we recorded. We measured the strength of adaptation by something called the adaptation index, which is simply the normalized difference of the early response minus the late response. And here in this scatter plot, you can see that the adaptation index generally gets larger during the active state. So adaptation increases. And we also looked at the speed of the adaptation. For this, we fitted an exponential to these response curves and looked at the tau parameter, which is the temporal decay. And when you look at the difference between this tau during the active and the quiet state, you can see that generally the temporal decay is faster during the active state, as I have shown you in the previous example, and you can also see in this example here. So we think that running boosts the processing of fast changes in visual inputs. Having seen this, again, we asked whether these same processes and effects would be visible in the retinal responses already. So for this, we again used NeuroPixels probes, but used an approach that was recently developed by another group. For the idea here is that you place your NeuroPixels probe parallel to the axons that come from the retina and then project to the superior colliculus. And if you get this right, you can see here how the action potential actually travels along the probe until it connects to a neuron and causes postsynaptic potential in a neuron. So here you can see the very specific shape of the spikes of these retinal axons. So Rehan and Florencia did these experiments and could collect data from several different retinal axons, and you always see this very specific shape of the spike waveform, which looks very different from the spike waveform emitted by typical superior colliculus neurons or any neurons really in the brain, which has fewer peaks and troughs. Okay, and you can also not see any traveling of the axon potential here. So with this method in place, we can really do the same experiments as before. We can record the spikes of one axon during the quiet state and again during the active state, and then compare these temporal dynamics during the different states. Again, you can see here two different examples. And so far, we have measured the strength of adaptation with our adaptation index. If you compare active versus quiet, we can see that even in the retinal axons, adaptation is stronger during locomotion, which is similar to what we see in the superficial superior colliculus neurons. Okay. The effect is actually even stronger in the deeper layers of the superior colliculus. So in summary of this, we saw that running also increases adaptation strength in retinal axons and presumably therefore also in retinal ganglion cells. To summarize this part of my talk, which is the main part, I've shown you that locomotion affects visual processing as early as in retinal ganglion cells, independently of any visual input, which means that information about locomotion is already present in the retinal signal. I've shown you that locomotion shifts tuning preferences towards higher visual speeds, which could boost the processing of fast visual inputs during running. And I've also shown you that locomotion enhances adaptation of neural firing to stable visual stimulation, which could boost the processing of frequent changes in the visual input. And from this, we concluded that neurons or neural networks close to the sensory input may already be able to distinguish between self-generated and externally generated sensory signals, and that the brain may use efficient coding by tuning its neurons specifically to the expected sensory signals. Now, I want to use the last few minutes to just give you a glance on a different topic we're also studying, namely the effects of reward on early visual processing. And this is important because reward marks relevant stimuli and features in the environment. It may be really beneficial to enhance the visual processing of these relevant features. To study this, we, and I mean myself and the master student Matteo Allemann, we trained mice in a specific visual decision task. So the mouse here either saw a stimulus to the left or to the right on the monitor, and its task was to move the stimulus towards the center by moving a steering wheel under its paws. And to make the task either more easy or more difficult, we changed the contrast of the stimulus. And here you can see a typical time course of a trial in this task. So at first, the mouse needs to hold the wheel still so that one trial starts. It starts by the presentation of the stimulus. After a short time, like a second or so, it hears an auditory go cue, which tells the mouse that it can now move the wheel. If it moves it to the correct direction, it receives a water reward. If it moves it to the incorrect direction, it hears an auditory noise stimulus. Then after another second, the stimulus turns off. And then a few seconds later, the next trial starts by another stimulus presentation. And I just give you a quick outlook of what I'm going to tell you. I'm going to show you data that shows that visual responses in the superior colliculus are enhanced to the stimulus presentation of the next trial if the mouse received a water reward in the previous trial. Okay, so if it received reward, visual responses are usually larger. So please keep this timeline in mind. And this effect you can see here in this example neuron. You can see the responses of the single neuron to different contrasts of the stimulus. As expected, the higher the contrast, the higher the response. This is after a negative trial, so the mouse did not receive any water reward, but if it did previously receive the water reward, the visual responses are much higher, as you can see here, and this is summarized in this contrast tuning curve here. When looking at the whole population, we found that about 20% of all superior colliculus neurons modulated it significantly its responses after previous reward, and most of these neurons enhanced or increased its visual responses. Okay, and this actually, yes, and we checked that this could not be explained by any other variables, for example, pupil-linked arousal, body movements, or longer reward history. So we made sure it's really the previous trial that matters for these responses to be changed. Okay. And this general increase in the responses had the effect that the decoding of the stimulus presence was generally better from the neural activity after the mouse received the reward. Here you see results from single populations, so single experiments. If we pool all the neurons across all experiments, we see a significant shift towards better decoding of the stimulus presence from neural activity. And interestingly, this was correlated with the behavior. So also after positive feedback, the mouse was much more likely to be correct than after negative feedback. Of course, that doesn't show any causal effect between the neural activity and behavior, but there is a correlation. And in summary, therefore, we think that reward may boost the processing of visual stimuli in rewarding contexts. So whenever you receive the rewards, whatever you see afterwards is better processed in the superior colliculus. And at least these effects, I think, should be considered when studying sensory processing in reward paradigms. So all these results, I think, bring up several more questions, and I want to finish with three of them here. So one interesting question, I think, is what the computational advantage is actually of these contextual modulations at such early stages of visual processing. So does the contextual modulation really underlie efficient coding of behaviorally relevant stimuli? Does it support the enhanced visual processing when relevant stimuli are encountered? Or is it just a byproduct without any advantages for processing? Another question that we tried or started to answer is, does contextual modulation differ across cell types in the early visual system? So does it support or suppress specific visual functions in the system? And lastly, I do also wonder how many different contexts and behaviors do have an influence on the earliest stages of visual processing, and how should we actually approach this question? Should we continue to test just all kinds of contexts we can think of, or maybe is it more fruitful to look at the possible underlying mechanisms and see what kind of information these mechanisms can bring to the retina or other visual areas? And at last, I want to thank first the members in my lab who collected all the data I've shown you in the later parts of the talk, as well as my collaborators that played a big role in the story that I showed you in the beginning of my talk, as well as the funders of this research. And now I'm happy to answer some questions if you want to.
Thank you very much, Sylvia. That was a fantastic talk. Really enjoyed that. Can I just start with one question? Yeah, maybe I didn't quite catch this. All that the modulation that you showed of locomotion. Do you think that all has to do with the histaminergic centrifugal input to the eye?
Short answer, no, I don't think so.
No.
I think this
Okay.
is just one mechanism. I think there are effects, and it has been shown
Hmm.
already that these axons exist and that they do have an effect on retinal ganglion cells. But I think that, I mean, we also know that there are neuromodulators projecting to the superior colliculus and that retinal axon terminals have receptors for those in the superior colliculus, and also the neurons have receptors, right? So I think, unfortunately, there's lots of things
Hmm.
happening here all at the same time. I
Hmm.
think it will be very challenging to figure out actually what each of these modulations do to the system and why. And yeah, t take all of this
Hmm.
apart.
And since you talked in the end about reward effects, is there any evidence for a dopaminergic modulation?
There is, but as far as I know, and last time I checked the literature, it doesn't come from VTA, so the typical dopaminergic area that we are thinking about usually, especially when it comes to reward. But there is input from the zona incerta, okay, that projects
Okay.
dopamine to the superior colliculus. But yeah,
There's a question from the chat that I now just read. On that, how do you think different neuromodulators play in locomotion versus arousal versus surprise versus reward?
great question.
Difference.
Um, Yeah, I don't really have
Mhm.
an answer. I think that, yeah, the
Mhm.
problem is there, even in the superior colliculus, a lot of neuromodulators are projecting there. There's also
Mhm.
serotonin, um, probably also histamine. So there's lots of
Mhm.
neuromodulators which also all have usually different roles, right? There's also, of course,
Mhm.
as noradrenaline, yeah. And
Mhm.
they all have
Mhm.
roles usually in locomotion and arousal and
Right.
surprise. So yeah, as I said before,
Mhm.
it will be
Mhm.
tricky to figure that out. So we need
Okay.
like
Mhm.
pharmacological experiments
Mhm.
to answer these
Yeah.
questions. At the moment, I'm not sure.
Yeah. And I know that in the much smaller world of bird vision, visual neuroscience, we are of course very mammalian centered, aren't we?
Yes.
Um, those centrifugal projections, from the brain to the retina, of course, has have long been known. Have we overlooked something in the world of mammalian visual neuroscience? Do you think? You know,
Well, actually, I think already Cajal saw some back
okay.
projections to the retina, so it has been known, but compared
Hmm.
to the birds specifically and also other
Hmm.
species, the back projections in mammals are far less in number. Um,
Okay.
so that each axon is from the data I've seen really has many projections, but there are very few axons, and it's really it's I think the experiments are quite tricky to do. Um, yeah,
So you mean few accents, but they contact a lot of target neurons. Are they very very sort of thinly spread out, so to speak? Right.
yeah,
Hmm.
exactly. So they have lots of
Hmm.
branches that go
Yeah.
that can
Yeah.
reach large parts of the
Okay.
retina. So even these
Right.
few axons may have substantial influences. Yeah. And
Hmm.
I just looked at
Okay.
some human data. So I think in the 80s, people already
That
tried
was
to
my
look
next question. Yes, you.
tried to look at this by recording like the electroretinogram. And but
Right.
it was debated whether like even attention. People were looking at attention, whether it has an influence on the retina, and the data I think was not super clear, but I think
Mhm.
people will pick up this topic again now. Also, maybe because
Mhm.
we saw things in the mouse, and not just us, right? Also,
Right.
several other
No, no.
groups saw these effects.
Mhm. Um, Is a follow-up question. Sorry if I missed this, but do your reward effects occur in trained animals, or are they there in training? Are they an error signal or a sign of increased attention?
Yeah, that's great questions. I guess I have to say that this protocol we used here was not designed for our question in mind. So basically, it was again a
Mhm.
discovery we had afterwards when we had the data. So, okay, to be precise, the data I showed you come from trained animals, but I always suspected that the training is not necessary because the effect
Mhm.
is right after the reward is received by the animals. And now in my lab, we have preliminary data showing that the same thing is happening in untrained animals. So as soon as you get the reward, the next visual stimulus that you see, I mean, in the lab, it's really easy. You have,
Mhm.
no stimulus, and then you have a stimulus. And
Mhm.
the next stimulus you see, those visual responses are enhanced. So training does not seem to be necessary. Um, so I don't think it's an error signal. I think it could be called attention, although I'm careful
Mhm.
with this term. Maybe something like, increased arousal, although this didn't show up in the pupil response, but something like alertness, arousal, attention. Yes, I think it's something like this. Yeah. I think terminology is a real problem. That's why I think it's a good idea to get to the mechanism so we can say, okay, this is the way this is happening, and then we can find a term for it.
Mhm.
Yeah.
Yeah, thank you. Um, I think there are no further questions at this point. So thank you very much again for a very stimulating talk, and food for further thought for sure.
Yeah.
Thank you
Thanks a
very
lot
much,
for the
Sylvia.
questions and for the opportunity.